first session and last session of today. I am Natia Hirankan. I will be the moderator for this last session. So this morning we have heard a lot about an exciting role of mRNA. But we already heard that that cannot be possible without, the use of LNP as a carrier, as a delivery into the cell and into our body. So this afternoon we are very fortunate to be able to listen to a real expert in the LNP. So let me give you a little bit of introduction. So it is my honor to introduce Professor Peter Collis. He is a physicist and biochemist, and I know that he's both a great scientist and entrepreneur. He has been co-founded more than 30 biotech companies, including Acutus, that have been, making the LNP that incorporated into the Pfizer COVID-19 vaccine. He is currently a professor at Department of Biochemistry and Molecular Biology, University of British Columbia. So please join me to welcome Professor Peter Collis.
Okay, thanks very much for the introduction. I'm a slow worker. I'm going to show you how it took me 50 years to get to where we are today. But it's a story really of my career as much as anything else. So I'm not going to go too much into the science. I'll do a few things, but I really want to sort of emphasize the journey of science as I've experienced it. So my title here is A Long and Winding Road to the COVID-19 Vaccine, mRNA vaccine, or how it took 50 years to develop these lipid nanoparticles. I have a number of conflicts of interest. It's not 30 companies. I have started one way or another more than about 10 or 11, but I think 30, that would be a little over the top. Okay. So you've all read these various headlines saying that the vaccines encapsulate or use tiny bubbles of fat. So this is just a headline from Bloomberg, but there's been many other press releases, etc., that say similar things. And in some ways, they're right. What are these tiny bubbles of fat? First of all, they are pretty tiny. They're 100 nanometers or so in diameter. Now, when you draw a line with a pencil on a piece of paper, the thickness of that line is about 10 cells wide. These things are about 100 times smaller than a cell, and so you can fit 1,000 of them within one line that you might draw on a page, just to give you an idea of scale. So we're really dealing with things that are pretty tiny and can get to most parts of the body. But they're really not bubbles of fat. They're made of membrane lipids, which are distinct from what we normally think of as being fat. This is, I'll describe a development since about 1972, where I got out of physics and then started to study membrane lipids. But I was involved not with anything that was applied. I was looking at very basic studies on membrane lipids, lipid polymorphism and lipidate symmetry, two things you probably never heard of before, but they actually ended up being integral to the COVID-19 vaccines. We spent a time between the middle 80s and the middle 90s, actually still going on today, to develop lipid-based nanoparticle systems, or lipid nanoparticles, to deliver cancer drugs more specifically to where they want to go. I mean, obviously, cancer drugs right now go all the way through your body and cause nasty side effects, and there's a real need to get them much more specifically to where they need to go. This resulted in a couple of drugs that have been approved by the FDA and the European Medical Agency to treat various forms of cancer. But since about 1995, we've been working on lipid-based systems to deliver nucleic acid-based drugs to target cells in vivo. And so this has again resulted in 2 drugs that received regulatory approval. The one of them, of course, being the vaccine, and the other being a drug to treat a hereditary disease. And I'll describe how that work evolved. So I got my PhD in solid-state physics. I was looking at semiconductors at 4.2 degrees Kelvin, which basically translates as transistors on the moon. It seemed like most of the interesting problems that I could see were outside the field of physics. But I did know quite a bit about nuclear magnetic resonance, and so, or called NMR. And so there were applications for that in the life sciences. And so I applied for a postdoc to go to Oxford, which I got to in 1973, but I knew nothing about biochemistry. So for any of you that get into a new field, this was a very daunting experience, going into a department where I was supposed to know something about what a protein was and what nucleic acids were, and I didn't have a clue. So I had to learn very quickly. But as I say, I did know a bit about NMR. This is my last physics paper, which I was very proud of because it had a lot of equations in it. So you really learn a technique in some depth when you go through a physics program. You also learn when you need an instrument that isn't available, you better build it. And the third is to go after some basic problems. So when I got to Oxford in 1973, we had a magnet, but not much else. And so that was my first job, was to build the NMR machine. Me and a graduate student put this NMR machine together. And that was, as I said, my introduction to biochemistry was not really much about biochemistry. It was about building an apparatus. Then I used the NMR machine that we built to study lipids in biological membranes. And as I indicated, the idea here was to go after pretty basic problems. So I got interested in the properties of lipids in biological membranes. Now, what you see here is a model of a biological membrane, and you get the lipids are the ones with the little balls on top and the long chains. And they play an integral role in membranes. They basically supply the structure and the permeability barrier for a membrane. And the proteins do a lot of the work, but the lipids are pretty darned important. So why are they interesting? Well, you've got about 30 trillion cells in your body. They're all surrounded by a membrane. There's obviously different forms of cells. But inside all those cells, there's various organelles. They're also surrounded by membranes. Membranes are probably one of the most important structural elements in a cell, and so there's good reason to be interested in membrane lipids. So this is just the sequence of, from a very complicated biological membrane to the lipid bilayer that holds the whole thing together, and then a membrane lipid, which has one end that likes to be near water, and the other end that likes to be sequestered away from water. So, but the basic message here, of course, is that membrane lipids are rather basic to life. There are people that say membrane lipids came first, and after that they trapped various reagents inside them to produce the nucleic acids, and so they're really the fountain of life as we might know it. Now, I got interested in the basic science of membrane lipids, and there's two instances here. Biological membranes contain, in addition to lipids that like the bilayer arrangement that supplies the permeability barrier, there's a lot of lipids there that don't. If you isolate them and put them in water, then they'll adopt non-bilayer structures. And of course, that can't be helpful to a lipid bilayer, at least one wouldn't think so. So the question was, well, why are they there? Also, biological membranes have asymmetric transbilayer distribution. In other words, the lipid composition on one side of the membrane is different than the other. It's called lipidate symmetry. And so what are the consequences of lipidate symmetry? So this is a long way from being a vaccine. So these are the kind of questions that actually, as I'll point out, led to some of the lipid nanoparticles that we're using currently. As I said, we constructed this NMR machine. We use phosphorus NMR. Phospholipids have a phosphorus atom. And so we could use phosphorus NMR to study the structural preferences of these lipids. And so you just add water to these systems. They'll spontaneously adopt their preferred structures. And we could detect that by looking at the NMR spectra. As you can see here, the bilayer lipids have a characteristic NMR spectra. The hexagonal H2 phase, as it's termed, which has these long tubes of water surrounded by the lipid, give a different structure, and I could go into all the physics in behind that if you want, but they did provide a nice signature, and it was a very useful technique to look at these structural preferences and what would cause lipids to move from one structure to another. And together with a postdoc that was in Oxford at the same time I was, we worked together actually for about 10 years, and published, must be about 70 or 80 papers on the lipid polymorphism side of things. We managed to explain using a shape hypothesis why lipids adopt one structure versus another. I will come back to this in the course of the development of the lipid nanoparticles, which is why I'm going into it now. But I'm pointing out how a lot of what we're doing now relied on these early stage efforts. So then we asked, well, what are the functional roles of non-bilayer lipids, of these lipids that don't spontaneously, like the hexagonal H2 phase? So what are they there for? What are they doing? Now, obviously, a biological membrane is pretty darn complicated, and so it's very difficult to do experiments on an intact membrane and look to determine what the physical roles of individual components are. And so what we used were model membrane or liposomal systems. And we developed a way of making these vesicular systems. They're shown on the right-hand side here, the cartoon, a cryo-TEM below that. But about 100 nanometers or so in diameter, maybe a bit less than that. We could easily make them. But we had to develop a way of making them, so we developed something we called the extruder, which actually turned out to be the start of one of our first companies, because we found that to get a rapid way of making these vesicular systems was something that was quite popular, and so the market was there, at least the scientific market. This is now one of the most popular techniques worldwide, in order to make liposome or univalvular vesicle systems. So we use these liposomes to show that membrane fusion and many aspects of life depend on membrane fusion, starting with fertilization, intracellular transport, cell division, many other things. But we showed these non-bilayer lipids really played a vital role there. And so if this and other functions that rely totally on the presence of these types of lipids. We also use them to demonstrate the consequences of lipidate symmetry. We synthesized a lipid, which we called an ionizable cationic lipid, which could exist either in a protonated form, which you see on the far left, which was, because it had the proton, a protonated form. This is a primary amine, as is indicated here. The lipid would then have a positive charge in the head group, or it could exist in a neutral form. And what we developed that lipid for was if it gets protonated, membranes are very impermeable to charged compounds. And so if this lipid moved to the inside, which it could do very easily in the neutral form, and then got protonated, then it would stay in the inside. And so we could drive lipidate symmetry using these lipids. So it was a great way. We could demonstrate huge shape changes as you flip a lipid from one side of a membrane to the other. And I'm sure that biological membranes use this kind of process to advantage for transport and other functional abilities. But we got distracted at this point, fundamentally distracted by the drug delivery potential. Now, in order to measure what we were using here was a pH gradient in the system, say pH 4 on the inside and so pH 7 on the outside. So the inside about the acidity of a lemon. So really not that acid, but you know, just a bit lower than, say, the physiological pH of pH 7. And we could use a weak base molecule. This is methylamine. That in the neutral form would go straight across the membrane, but in the inside get protonated in the lower pH environment. And at equilibrium, the concentration on the inside versus that on the outside would mirror the proton gradient. So in other words, the pH unit, say 3-unit pH unit, be 1,000 times higher concentration of the weak base on the inside of these lipid nanoparticle systems. Then we noticed that many common cancer drugs are weak bases. This just shows about the 4, but many of them are. And so we reasoned that maybe these could be taken up in the same way, and it turned out that was the case. That we could load these little liposomes, model membranes, with cancer drugs to such high levels that they would precipitate inside. The bar there is 100 nanometers, so we have little nanocrystals forming on the inside of these lipid nanoparticles. And so the immediate, what we did at this point, we went through a number of iterations, but we started a company called Inex Pharmaceuticals to use these liposomes to improve the delivery of cancer drugs to tumors. I mean, this is a huge problem. Right now, when we use a cancer drug in the clinic, the less than 0.001% actually gets to the tumor. The rest goes right through all the way through your body and obviously causes all kinds of nasty side effects. And so we need drug delivery systems to take these drugs much more specifically to where they're needed. Half of my lab is still working in this area of trying to get much more specific delivery, and we're getting closer. It's a huge issue. So we formed this company by me and 4 postdocs in my lab. This is just indicated here. And part of this was really to keep a team together. We had a great group, and it certainly was this is one of the things I'm most proud of, is that this group of people that were working together 40 years ago, and many of these people I'm still working with today. And it's the reason for our success in the long term that we managed to keep this group together. So as a result of these kinds of studies and the companies that we formed, we got 3 drugs approved to treat cancer and its complications that received regulatory approval. So, this is really just demonstrating from a lipid nanoparticle point of view, a lot of the technology that we're using now is not new. I mean, these drugs were approved in 1995, 2000, et cetera. So the reaction of the body to the presence of these kinds of drugs has been well studied. But forming Inex was really vital, because in order to bring a drug to market, you need a lot more than just good science. In this case, as I mentioned, we needed to keep a pretty good team together. Need to raise a lot of money, because doing the clinical studies is hugely expensive. We were paying $50,000, for example, per patient that we were treating in a clinical trial. And so 100 million or more was the kind of money that was necessary. But you need also different forms of expertise, of financial, manufacturing, regulatory expertise, clinical expertise, obviously, business development and deal making. The list goes on and on. So one of the consequences of being in a company, though, is that you sort of have to be sensitive to changes in direction. And we'd started, this is the mid-90s, we'd started INEX to deliver cancer drugs, as I mentioned. But one day the CEO came to me and he said, I can't raise money putting old drugs into these liposomes. We need to be doing gene therapy. It has, and this was the exact words, he says, has much more sex appeal. So at least it did for investors. So it started us off really on a whole new path. And this is getting more to the crux of the matter in terms of the LNPs for vaccines and other applications. Now, what is gene therapy? Wikipedia has a very general definition. That's the therapeutic delivery of nucleic acid into a patient's cells as a drug to treat disease. But the operative word here is delivery. They need a delivery system for a variety of reasons. One of those, or three of those, are that these molecules are very easily degraded. They don't get to where you want them to go when you inject them into your body, and even if they get there, they can't get across the cell membrane to do their work on the inside on the interior of cells. And so the delivery system here is crucial. Now you remember the central dogma is this term of molecular biology, which is DNA makes RNA makes proteins. And so we have to either prevent the translation of pathogenic proteins or encourage translation of therapeutic proteins, and this is using nucleic acid polymers. This is the practice of gene therapy. Now, we focus primarily on RNA-based molecules because they can either inhibit gene expression or promote gene expression without involving the genomic DNA. And of course, that has advantages from a safety point of view. One can use short interfering RNA to combine with the mRNA or to get the mRNA coding for a particular sequence or a particular protein to be degraded, or you can use mRNA to cause the production of therapeutic proteins. But of course, both of these are large molecules that can't get across cell membranes on their own, and so they require delivery systems to reach the interior of target cells. So since about 1995, we've spent that time developing these LNP systems to deliver small interfering RNA and mRNA into cells in vivo. So I'll just go through a bit of the design of these systems, talk a bit about the Onpattro story, and then subsequently the BNT162b2 or the COVID vaccine story, the Pfizer-BioNTech COVID-19 vaccine. So our aim in 1995 was to develop a delivery system to get siRNA into hepatocytes in the liver to silence the gene in the liver. This is a pretty huge challenge. I mean, as you're aware, the immune system is exquisitely tuned to make sure that no nucleic acids get inside cells. For obvious reasons, of course, viruses have evolved to try to avoid this, and that's of course one of the reasons why they have certain pathogenic effects. That's the reason why we have such elaborate immune systems. And so what we're trying to do here is package up the nucleic acid, inject it intravenously, have it go to where it wants to go, get out of the circulation, get taken up by a cell, and then get out of the endosome, as it's termed, to get into the cytoplasm. This is quite a trick in order to design systems that might do this. Now, the first problem that we hit was that efficient encapsulation of the negatively charged nucleic acids requires a positively charged lipid or cationic lipid. Now, there's no permanently positively charged lipids in nature. They're either neutral or they're negatively charged. Positively charged lipids are really toxic. They'll tend to rip membranes apart. So this is a big problem. How do we encapsulate these polymers without having this toxicity? And we learned the hard way that with, say, cancer drugs, anything that adds to toxicity, you got a big problem. So we went back to the work that we've been doing on lipidate symmetry, and as I mentioned, we'd synthesized this lipid called DODAP, ionizable cationic lipid that could exist in a positive form or a neutral form depending upon the pH. And we asked the question, well, what, if we lowered the pH to pH 4, and where it's positively charged, could we load nucleic acid polymers into a lipid nanoparticle, and would the nucleic acid be retained when we brought the pH back up? So a pretty simple question, but if we hadn't had the ionizable lipid as a result of the basic studies, we would have never thought of doing this. And so we developed a process where you dissolve the lipids in ethanol, and in this case it was either DNA or RNA, small segments of DNA and RNA, antisense oligonucleotides, etc. Dissolve those in water at pH 4, and then rapidly mix the ethanol, the lipids in ethanol with the water containing the oligo. And you can see intuitively here what could happen. One of the first things to fall out of the solution will be the negatively charged polymer surrounded by the positively charged lipid. And if you do it fast enough, you can have other lipids in there. And this is like this thing with a squiggly top to it, the PEG lipid, the surface lipid. If that falls out of solution subsequently, it'll trap these systems in a limit size particle, as we term it. In other words, the smallest size particle that's compatible with the molecular components. And so and then after that, dialyze away the ethanol, raise the pH, and you've got your finished lipid nanoparticle. So this was the situation we arrived at fairly around the year 2000, something like that. And it really is an ideal process for encapsulating RNA or DNA in a lipid nanoparticle. They're relatively non-toxic because they're not charged at physiological pH values. The hydrophobic core, stable encapsulation efficiencies of pretty much 100%. And we can adjust the size by just simply changing the ratio of the surface lipid to core lipid. So they're quite flexible processes. So that was the start. I mean, you'll notice how we didn't go straight for trying to get something to do something in vivo. The first thing we had to do was get something that could potentially be a viable product in terms of encapsulation efficiency, toxicity, and those very basic reproducibility, those very basic parameters. One of the things that a lot of people tend to do is to develop a system that's not well characterized and then go straight into an animal model, and then you have a problem because if you're not producing the same thing each time, then the number of variables can be overwhelming, and it's very difficult to make progress. So now I'll talk a bit about the Onpattro story, which started around 2005, and this was developing a drug to treat a condition which you've never heard of called hereditary amyloid transthyretin amyloidosis, which affects about 50,000 people worldwide, and it's pretty nasty. And so I'll just go through that as the next step. So this is siRNA. The small molecule drugs, like cancer drugs, have a molecular weight of about 500. siRNA has a molecular weight of 13 kilodaltons, 13,000 molecular weight. So this is a big molecule in comparison to most therapeutics. So here I was at a conference in London. And I was pursued by a guy named Mad Russian, Viktor Kotelyansky. He was the VP research for a company in Boston called Alnylam Pharmaceuticals, who had been founded 2 years earlier to use siRNA as a therapeutic to inhibit the production of proteins that were causing deleterious effects. And he said, we have a problem. How do we get our siRNA into the liver, into hepatocytes in vivo. And is there a delivery system that you have that we can take advantage of? So this resulted in a really very productive collaboration with Alnylam that went on for about 7 years to develop these LNP sRNA systems to silence genes in the liver. So it's involved INEX and subsequently Acuitas Therapeutics, as well as my UBC lab. This is the two teams. We had 8 chemists working continually over this period, 300 different lipids, testing many formulations, and it resulted in a very viable product. So these are the simple systems that we developed as a result of using the ionizable lipids. This is a cartoon rendition. So we said, okay, can we silence a gene in the liver using these systems following an IV injection? Now, as has already been alluded to, these lipid nanoparticles get into cells by a process called endocytosis. And so our job is to get these lipid nanoparticles to break out of the endosome before it goes on to what's called the lysosome, where everything's going to get degraded. So there's a critical point here where we want to get the maximum release of whatever the cargo is. So this is the objective. Now, how do we get ionizable lipids to destabilize endosomes? Well, we knew that permanently positively charged lipids could destabilize membranes. And so we started to ask the question, well, why is that? How does that work? And what we found was that if we took these cationic lipids and added them to the negatively charged lipids we find in membranes, they flips them right over from a bilayer structure to a hexagonal phase, these non-bilayer structures. This accounts for the toxicity, of course, because it just rips biological membranes apart. So the trick here was to say, okay, can we design these ionizable lipids so that they can take advantage of this sort of process to destabilize the endosomal membrane? So we had to design them so that they would get to a certain point after being taken up. You'll notice that the pH in the endosome goes down as the endosome matures, and so that these ionizable lipids will get, because they're neutral at physiological pH, but they're charged at lower pH, they'll get protonated and become cationic and able to combine with the negatively charged lipids as they progress in this pathway. So we're back to lipid polymorphism, this very basic work that we'd been doing 20 years before. And so we designed these ionizable lipids, both in the sense of their shape in order to get the maximum non-bilayer preference, but also their pKa, the pH at which they became protonated. And this ended up being quite a useful approach. So we used a factor 7 model. So factor 7 is a clotting protein that's made in the liver. Obviously secreted into the circulation. So we designed an siRNA to silence factor 7 and package that in a lipid nanoparticle, inject these mice via the tail vein, and then assay for factor 7 in the blood some 24 hours later. And so if the more potent these systems are, the less factor 7 we're going to detect in the blood after injecting them with these systems. And what we found was that the potency was extremely sensitive to the species of the cationic lipid that we employed. We went through a lot of different formulations, but varying the pKa, varying the pH at which they became protonated, they were highly sensitive to that particular parameter. These are a few of the lipids. They're pretty simple lipids in terms of the structure, and they all have this amine head group, primary amine, secondary amine head group, dilenoyl acyl chains, but their activity is dramatically changed according to the pKa. So this is just indicating the potency of these systems, where we're defining the potency as the effective dose that we need to get 50% gene silencing in the liver. And as you can see from this, the y-axis here is a semi-log plot. If we move as little as half a pH unit away from some optimum, we can be reducing our potency by as much as a factor of 100. And so this was certainly an unexpected benefit of these ionizable lipids, that they have this property that we could start to tune them so that they'd be able to engender the breakdown of the endosome and the release of the nucleic acid cargo. So this just indicates where we started. We needed 10 mg per kg, and we ended up at about 5 micrograms per kg. And if anything, the toxicity of these systems was reduced. And so we had this huge therapeutic window that we could give 1,000 times higher dose before we would start to see any toxicity arising from these systems. This is in a mouse model. But it just points out you need to start with something where you have a huge therapeutic window in order to have a chance of getting a viable therapeutic. And so this was pretty, around about 2011, somewhere in there, we arrived at this point. Now, if we could silence factor 7, we can silence any gene we want. This is the nice generality of a gene therapy approach. We just have to get a different siRNA, and then we can silence whatever we want. And so the clinicians at Alnylam said, okay, well, let's go after this transthyretin-induced amyloidosis disorder. It was actually a very good decision. So transthyretin-induced amyloidosis, this is really a multisystemic disease. It's caused by deposits of transthyretin fibrils. You can get a number of mutations of this protein. It's made in the liver, secreted into the circulation, and it transports it's a retinol binding protein. But if there's some of these mutations, then it can end up forming these fibrils, which presumably distribute all the way through your body. But it really they really have nasty effects in nervous tissue and in cardiac tissue. So there's no effective therapy, and it's usually fatal within 5 years of diagnosis. And this just indicates the progression of the disorder as one progresses through the various stages. Obviously, a very nasty disease, and because it's a hereditary disease, if you started stumbling, which is one of the first indications you've got a problem, you would know that you're in for a pretty nasty sequence of events, because you'd likely seen parents or uncles or other relatives who had gone through the same process. Now, one thing about the gene therapy approach is that the hypotheses here can be remarkably simple. If we shut down the production of transthyretin, maybe this is a way of treating this disorder. And so the idea here is to use transthyretin, use siRNA to knock down transthyretin, and maybe then we'll reduce the number of fibrils that are circulating. The deposition will be reduced, and perhaps even we'll get clearance. So it's potentially a simple solution to a devastating systemic disorder. So the first phase 1 studies was done in healthy volunteers. This is around about 2013, and came up with some quite spectacular results in terms of gene silencing in people, and showed that at about 0.15 milligrams of siRNA per kilogram body weight, you could get more than 80%, 90% reduction in the levels of transthyretin in the circulation, which lasted for 2 or 3 weeks. So a dose of 0.3 mg per kg of siRNA every 3 weeks was chosen for the subsequent clinical trials. The trial involved 225 people, 148 getting the drug, the LNP sRNA to knock down transthyretin, or 77 got placebo, which was sterile saline. The endpoints here were a neural impairment score, ability to walk, a number of other quality of life, self-reported, et cetera, et cetera. Now, this was, over this 18-month study, some pretty spectacular results were obtained. The neural impairment score for those on the placebo group, which is in purple here, proceeded to get worse over this time period. But the people that were getting the drug, the sRNA to knock down transthyretin, if anything, got better. And so one's not only stabilizing a hereditary disorder, you're actually, these people are improving. So pretty remarkable. The clinical trial results that were announced in 2017, and they really hit their primary endpoint and all of the secondary endpoints. I don't know why they bothered to calculate the p-value here. This drug clearly works. I often say this is 1 over Avogadro's number, so it's a pretty convincing p-value, as one can see for the other many of the other secondary endpoints: muscle strength, nutritional status. As you can see, this is a wasting disorder, so it's a stabilizing and potentially curative therapy for a previously fatal disease. So I already thought this was going to be the apex of my career. It's approved by the FDA in 2018 for treatment of H8 of transthyretin-induced amyloidosis, the first FDA approval of an sRNA-based drug and clinical validation of the lipid nanoparticle delivery system. So I'm just going to go on now to the story post 2012 when the drug. When Onpattro went into clinical trials, obviously there's not much else that one can do as a, in drug development, because if you change the drug, then you have to start the clinical trial process all over again. And so at Acuitas, we went on to say, okay, well, if we can deliver small interfering RNA, 13,000 molecular weight, maybe one can deliver messenger RNA and get gene expression in the liver in the same way that we were delivering these big molecules to the sRNA to the liver. Perhaps we can get mRNA to the liver as well. And so I'll just briefly go through that in the next few minutes. So does it enable mRNA-based gene therapies? And so we wanted to find out. So this is cartoon or diagram is exactly the same as the one for siRNA, only now we're packaging something that's hundreds or more kilodaltons inside these lipid nanoparticles. Now, if we use the lipid called MC3, which was the one that was the optimized lipid that we had for the lipid nanoparticles that for Onpattro, we could see, using this IVUS imaging approach, we could see protein production. This is luciferase expression in the liver after an IV injection. So this gave us some considerable indication that the approach might actually work. And so Acuitas has gone on to really say, okay, well, what can we improve on MC3? So MC3 is the little one on the left-hand side here in terms of relative luciferase expression in the liver, and you can see now getting up to 20 to 30-fold improvements in gene expression levels in the liver. So the ionizable, this is mainly ionized changes in the ionizable lipid. It certainly is one component here that just keeps on getting better. There's a huge number of applications here because we can now use this kind of approach to produce any protein we want, just using the liver as a bioreactor. And so this you can get supramolecular or supraphysiological levels. If you want to win the Tour de France, then this is another way to do it. This is for EPO, erythropoietin. So a single IV injection, really, this is in pigs, in a pig model, so a large animal, but showing the kinds of increases in the circulatory levels of the this hormone that can be achieved by an LNP mRNA system in this case. Here we got, this is where if you don't have good luck, then you've really got a big problem. So we were approached in 2014 or around that time by Drew and Katie Carrico, who had done, as we're all aware, many years of work developing mRNA for vaccine applications. But in the same way as when we were working with Alnylam, we have a problem. How can we get our mRNA into muscle and immune cells in vivo? So we weren't going in this direction at all. We were obviously trying to get better and better systems for expressing proteins in the liver. I don't have to go through this in any detail because it's been covered in the 2 previous talks by people who are much more expert than me. But in terms of the immune response that we needed, that was desired, MHC1, MHC2 class immune responses as a result of injecting these systems intramuscularly. Now, Drew already covered off on the influenza results. This is just other results that came out of Drew's laboratory for Zika virus. And so this is one of the first, in addition to influenza, that we looked at. And so here we have mRNA that's coding for the Zika virus pre-membrane and envelope glycoprotein, and packaging that in the lipid nanoparticle and injecting that, I think that was intradermally actually. But the results here just demonstrate again the potency of these systems as vaccines. So injecting intradermally on day 0, and then challenging at 2 weeks or 20 weeks with the live virus. And showing here that there's total protection in these animal models, both at 2 weeks and at 20 weeks after immunization. So these results looked pretty spectacular and caused the association of Acuitas with BioNTech. The partnership there was again focused on influenza actually. And in turn, BioNTech had been working with Pfizer on a flu vaccine, and you've heard this story a little bit already. So of course, all efforts switched from a flu vaccine to a COVID-19 vaccine in January of 2020. I was asked to go down to Pearl River, Pfizer's headquarters, to give a talk. I guess that was in February of 2020. And I really couldn't understand why it was. I had senior people from the company in the audience. Because usually when you go to a pharmaceutical company to give a talk, well, you're talking to the people at the bench, the scientists, et cetera. But there were some pretty senior people there, and I didn't really figure that out. Until I realized that they were about to make a decision to actually move forward with this into the clinic. So yeah, serendipity certainly worked well here, that all these things came together. Here we're, of course, putting in the mRNA coding for the SARS-CoV-2 spike glycoprotein into these lipid nanoparticles that had evolved through this complicated path that I just went through. And you all know the results of this. These are results that were announced November the 18th of 2020, demonstrating 95% effectiveness for preventing the SARS-CoV-2 virus, the COVID-19 disease. Consistent right through age, gender, ethnicity, etc., and also pretty well tolerated. Again, as you've heard, there's 44,000 people in this trial, which is a really big trial for a vaccine. And so the confidence in this was pretty strong. So this has been approved. This is an older slide. It says up to 1.3 billion doses by the end of, well, actually it's a press release. They said they were going to get 1.3 billion doses by the end. I think it's ended up at 3 billion or something in that realm. It points out actually how the manufacturing process here is readily scalable. It has been approved in many jurisdictions. So we spent this time, these 25 years, and as you can see, I've tried to trace through the zigzag path on the way there that has really evolved into what is, right now, I think it's the most successful new drug in history in terms of the number of doses and the impact, the worldwide impact. But really, the vaccine applications, as we've heard, are just the tip of the iceberg. There's many other gene therapies that are going to be enabled here, and we've heard what a large number of those are. Again, I would say to people in the audience that the younger generation of scientists, this is a fantastic area to get into, because there's really so much that can be done now that we could never do before using this kind of approaches. And certainly cancer, heart disease, brain disorders, whatever, are now becoming much more attackable than we might have thought. The one thing that I find fascinating is the way in which the potential for really personalized therapies, where so this is simply because things can be done so quickly. Once you identify the protein you want to make, or you want to edit, or you want to silence, then you can produce the sRNA or mRNA in a time on the order of weeks. And the encapsulation takes a day. And so you can have these highly personalized therapeutics that are available in the time frame that's really quite useful. For example, if a child is born with some genetic deficiency, which if they don't get the right protein, then they may well die. Well, these are things that can be done in a very, I'm going to say very straightforward. It's not that straightforward, but the potential is that it can be done. So all of this arose from very basic research on the roles of lipids in membranes, and I always like to give some sort of a boost to basic research efforts, because if you don't have this basic research effort going on, you don't have the tools to really enable you to make advances in therapeutics. So there's a number of lessons here. The basic research one I've just indicated, lipid polymorphism, lipidate symmetry, don't conceptually have much to do with gene therapy. Starting companies I found to be a very useful way to go forward, to keep a really good team together. And this is true with INEX and with Acuitas. And the collaborative nature of science that is also vital. Without the work with Alnylam and with Drew, we would never have got to where we are. So I'll just close with a few acknowledgements. As I mentioned, I've been working with people like Mick Hope and Tom Madden for 40 years. The many others, Steve Ansell, Ying Tam, Barb Mui, Paula Lin, Sean Semple for 20 years or more. And it's those long-term relationships that have really made this a fantastic journey. Alnylam, Mark Tracy, Kenneth Kink, and Martin Mayer, these were formative moments. My own group, and of course working with Drew at the University of Pennsylvania. So it's been quite a what's the word? A fascinating evolution that I think points out how science is very difficult to be predictive of what you actually need in terms of developing new approaches to therapies. And in this case, things have fitted together very well. So with that, I'll take any questions you have.
So thank you very much for such an inspiring and impressive work. And I really love your lesson of slide. And because I'm teaching basic science, I think that's an excellent take-home message here, that basic science for medical student and all the science student is the most important things. And taking this real-life example here, that he started as a physicist, but now he's like more expert in cell biology than any of us. So thank you very much. And are there any questions from the floor?
Hello. Who is Ian McLachlan, and how is his platform for the lipids related to yours work?
Sorry, what's the question again?
Who is the Ian McLachlan?
Oh,
and
Ian,
how is his
yes.
discovery related to your research?
Right. So Ian was I hired him into Inex Pharmaceuticals, and he was helping us with the gene therapy approach. Now, in the year 2000, we started to get some good results with our liposomal formulation of an anti-cancer drug, and we put more effort into that. And so we spun out some of the gene therapy work into a company called Protiva. So another company I co-founded. So he was the chief scientific officer in that company.
Yes. But so what I catch from the Forbes, he is the one who developed the platform for the now that is used in the vaccine. Is it overly simplified version, or is it really true?
I don't know if I quite catch your question.
So based on the interview in the Forbes.
They definitely made a contribution. At that point, we had the ionizable lipids, we had the pegolipids, we had the cholesterol and phosphatidylcholine and all of that in there. We were working on antisense oligonucleotides, and Ian's group moved that on to siRNA, working with Alnylam, and so they managed to get that system working for siRNA, which is a more potent molecule for silencing genes. So,
yeah, thank you.
Hello. Okay. I have two questions. So one of the organization work on the cancer vaccine. So he tried to adjust the proportion of cationic and anionic to try to target like spleen instead of lung. Can you explain that? How does it work?
Yeah, you're talking about James Dahlman's work, right? Yeah, you can certainly, what he's shown is you can get different distributions to different tissues. I'm always a bit leery when people are using these cationic lipids. The potential for toxicity, I think, is quite high. The other part is, the proportion of material he's getting to those sites, is not huge. I suspect that approaches such as Drew are using an antibody approach, or as approaches that we're pushing, where you use some of these natural targeting ligands, will end up being more productive. But that's just my view. It may well be that some of these other approaches are viable. That we're using these lipids with various charges. But the toxicity issue is one that always worries me.
So in your view, in order to target the specific tissue, better to use like affinity-based instead of
Sorry, I didn't quite catch that.
In your view, in order to target specific tissue or cell, what is the best approach that you think?
I think it's going to be possible to do that. There's a number of ways that one can go after it, though. One of the things that we're doing is the systems that have been developed to this point are they don't circulate for very long. Within 30 minutes, they all go to the liver. And so the trick is to get long circulating systems that will circulate around for hours, and then then they'll reach these other tissues, and then find ways of turning them on or off according to where you want the gene therapy to occur. And so I think that's going to be that's one of the nice things about the flexibility of these lipid nanoparticles. that you can take along other things. I mean, for example, you can incorporate gold nanoparticles or other things that are sensitive to external radiation. And so I think that some of those physical approaches are going to be like the first step of getting gene therapy in a particular area. So it's again, these are, from the lipid nanoparticle field right now, there's a lot of, as you can imagine, a lot of experimentation. We now have long circulating systems, so they get pretty well to the bone marrow, and so the possibilities there from a therapeutic point of view are breaking open. And similarly for spleen, well, as Drew was also indicating, for hitting T cells and other immune cells. So these are, this is where the literature now is expanding, daily, in terms of the, getting new tropism for different tissues.
I can have one more question. Can you compare those exosome, EV, versus the LNP? What is your opinion on the advantage and disadvantage? Okay.
Yeah, the exosomes are that's a difficult technology. In terms of loading them with cargo and having reproducibility in the exosome itself, they're pretty complicated, and the manufacturing process is not straightforward. They certainly have some advantages in terms of being able to access, tissues. I'm not convinced yet how useful they'll be. They seem about as complicated as a virus to me, so I would, the simplicity of the lipid nanoparticle is very attractive.
I have two questions. Does the lipid nanoparticle be able to deliver bigger molecules such as plasmid DNA, let's say the Sleeping Beauty or piggyback transposon, for more stable expression of the interested protein?
So you're asking whether or not they can deliver plasmid DNA or bigger molecules? Yeah, there's really no limit to the size of the DNA or RNA that you want to put in. What happens is that when we're working with siRNA, the number of copies of siRNA we'll have per lipid nanoparticle, maybe it's 60 or 70 nanometers diameter, we'll have about 1,000 copies of the siRNA per lipid nanoparticle. When we're dealing with say a mRNA that's a few hundred kilodaltons, then you might only have 2 or 3 per. But then when you go even higher, you may end with just 1. But to the size, because it goes as the cube of the radius, you can accommodate. It just gets a little bit bigger, and maybe there's less copies per nanoparticle, but it can accommodate all of those types of systems.
One more question. Comparing with the core polymeric nanoparticle, what is the advantage or the key benefit of the LNP over the core polymeric nanoparticle?
The advantage of the LNP over what?
The core polymeric nanoparticle.
Well, one works and the other doesn't. I'm just kidding. The lipid nanoparticles are certainly much more effective at getting out of endosomes and into and delivering the cargo to the interior of cells. There are, as Drew was indicating, from a vaccine point of view, there seem to be profound advantages in terms of the adjuvant effect, which you don't get from the polymer. So that's another reason.
Thank you so much.
Thank you very much for your excellent lecture. I am Rangsini from Chulalongkorn Dental School. And at the dental school, we try to develop the therapeutic mRNA for periodontal tissue regeneration in periodontitis patients. So we need a good scaffold for the N1-methylpseudouridine modified mRNA encoding growth factors in LNP, which kindly provided by Professor Drew and Dr. Norbert Pardee. So we're working on the animal model, a rat cavalier bone defect for bone regeneration. Would you like to share your information about scaffold and the LNP? At the dental school, we use the collagen scaffold, which everyone seems to use that kind of scaffold.
Yeah. Yeah, we really haven't done much work in that area. I do think it's, I mean, there's a couple of things you're going to need to do. One is the retention of the lipid nanoparticle in the scaffold, and so the ways in which you can make it have affinity for that scaffold and then pay out over time. These are very doable things, I think, but you do need a combination of expertise on the polymer side or the scaffold side in combination with the LNP. I mean, one of the ways to go with the LNP side is to make systems that have particular things on the outside, not necessarily charged, but things that will bind to agents that will bind to material on the scaffold. And so it's going to be quite specific to the particular scaffold you want to use. As I say, we really haven't done much work in that area, but I think it's entirely possible to do, and then have a slow payout of the LNP with the mRNA coding for whatever you want to do from a bone regeneration point of view.
Right. So our LNP is a neutral charge, right? And the collagen, and we're trying to use a variety of scaffolds such as the silk fibrin or hydrogel or chitosan, right. But we know very little about the properties of this scaffold. We would like to recruit all the bone cells or osteoblasts to come into the scaffold and get transfected and control release of the proteins, things like that.
Yeah, yeah. As I say, I think it's very possible to do. But each one is going to be quite a specific. If you want to get the fibroblasts coming in, then reprogramming them into whatever. There are definitely efforts being made to do that. Yes. And I think we're going to see lots of making endothelial cells for bone, for wound repair and things like that. These are all going to be coming along quite quickly, I think.
Right. Thank you very much.
Yeah.
Hello. Well, congratulations. It's really been an amazing journey. I have a practical question, but I'm not sure whether it's you or Professor Weissman who would be an appropriate person to respond to this. After we get Pfizer vaccine, especially young men like me, there are a higher chance, relative, well, not high, but relatively high chance of getting myocarditis. And governmental agencies recommend that we should restrict our physical activity. Do you think that's rational, that's helpful, and should we do that? And why? Is it because
I think Drew covered this with the statement that I don't know if you get COVID, the disease, you're 30 times higher chance or whatever the number is of developing myocarditis than if you than from the vaccine. It's probably a bigger ratio than that. So I don't know, Drew, if you want to comment on this any further, but I think the message is that you're in much better shape to get the vaccine. The incidence of the myocarditis seems to be vanishingly small.
Yeah, we are getting. I have got Pfizer. My son's got Pfizer. My question is, should we restrict our physical activity for a week after we get vaccines? Is that rational? Is that helpful?
I don't know. I mean, I'm not a doctor, so I just don't know.
Okay. Thank you.
Yeah, maybe you have to wait until Drew publish the reason why we're looking forward to that.
I'd like to have, so could you touch on the thermostable, whether if you want to improve the stability of the mRNA with lipid nanoparticle, what the role of the lipid nanoparticle or formulation can improve, stability of the mRNA in a higher temperature.
Right.
Yeah.
I'm not really close to those studies. I mean, as I've said in other venues, a lot of the basic studies that were on stability studies, they didn't do those because they moved this drug into clinical trials very, very quickly, within 3 months, and didn't do couple of years of stability work to get adding various excipients. As I understand it, the minus 80 requirement came in because if they went to minus 20, the lipid nanoparticle froze very slowly, and there was some physical disruption to the LNP that they didn't observe at the minus 80 storage condition. I think they're relaxing some of the requirements now and finding that they can store it for longer at a much more elevated temperature. But there's no intrinsic reason why we have to, well, certainly not for minus 80. I mean, I think in the end we're going to see refrigerator temperatures as being adequate for a year's stability. The LNP for sRNA, for the Onpattro product, that has a year's stability in an aqueous form. Whether we can achieve that for the mRNA formulations, I don't know, but I don't think there's any intrinsic reason why not.
Well, we are right on time. Well, thank you very much, Professor Peter Collis, and I would like to invite Professor Kiet to give a Thai souvenir for Professor Peter.
Yeah.
Thank you, and on behalf of the ChulaVRC, Faculty of Medicine, Chulalongkorn University, I would like to thank all the speakers and all the audience, both on site and online, for being with us all day. And I hope most of our young scientists and medical students get inspired by real story. And again, thank you very much, all the professors, for coming here and share with us your wisdom and real experience. Thank you very much. อาจารย์เกียรติจะปิดหน่อยไหมคะ
No, I think thanks again for everyone, and thanks for all your great speakers that spent the whole day with us. So we have another hour for internal discussion. So thank you very much, and hopefully we're going to have another forum next year, and hope all of you will join our forum once again. Thank you very much.